Hey everyone, this is Elad from Astrolab Diagnostics, and this video is part of a series about immune monitoring, specifically the role of flow and mass cytometry in the context of this groundbreaking technology. As with the previous video, full disclosure, I am neither a biologist nor an immunologist, so if I make any mistakes, please feel free to reach out and let me know. In the previous presentation, I was talking about immune monitoring, and the idea with immune monitoring is that we track the different cell subsets in the immune system, examine their frequencies, their states, and the interactions between them in order to learn something about the progression of a disease in a given patient or a set of patients. I also mentioned that under the umbrella of immune monitoring, we can find multiple different technologies, such as single-cell RNA sequencing, ELISA, ELISpot, Luminex, and flow and mass cytometry. For the context of these videos, I'm going to focus on these latter two technologies. Flow and mass cytometries are sister technologies, and while they do differ in their chemistry and in their physics, the end result is quite similar. So I feel that we can confidently bundle them under one term, just for the context of these discussions. In the context of flow and mass cytometry, immune monitoring is the ability to robustly identify cells from the different immune sublineages and stratify them into different subsets. Then, for each one of the sample in our experiments, we would like to know what is the composition of these subsets for that sample. And to provide some more context on this statement, we need to take a step back. As I mentioned in the previous video, the immune system has different cell subsets in it, and they have a variety of roles. Some of them identify pathogens, some of them destroy pathogens, some of them allow our bodies to adapt to pathogens to better identify and eliminate them, and some cells in the immune system regulate the rest of the cells. Furthermore, it is quite common to see cells with different functions or a combination of functions, and this explains the large heterogeneity that we see in the immune system. Now, broadly, there's four major categories of immune system, and these are B cells, T cells, NK cells, and myeloids, which include dendritic cells and monocytes. However, this is quite a broad overstatement, since there's a lot of distinctive populations and there are some cells that don't fall as nicely into these major compartments. So there are details that I'm skipping here just for the purpose of keeping this presentation accessible. So apologies to any granulocyte or ILC biologists out there. Cell subsets can be identified by their expression of specific proteins, which are referred to as markers. These markers can be found on the cell surface or inside the cell. Now, there are a lot of these proteins that can be expressed, and of course there are cells in our bodies which do not belong to the immune system and express their own markers. So there is a dictionary or different rules that people use based on prior research in the field. And this is one common example, one very common hierarchy of classifying different cells in the immune system. So we can see that in this hierarchy, there's different types of T cells, different types of B cells, and different types of DCs, monocytes, and NK cells, with quite an emphasis on T cells. However, this is just this hierarchy. And just to zoom in on a few of the sections in this plot, we will see that T cells are known to express CD3, B cells are known to express CD19, monocytes express CD14, and DCs and NK cells are negative to all of this and positive for their own markers, which are denoted down here. I will point out that there's always exceptions to these rules. For example, earlier B cells, younger B cells don't express CD19, or not all monocytes have CD14 on them. There are CD14 negative monocytes. Furthermore, we can use these markers to measure the cell state or behavior. One notable example is whether a T cell is activated or exhausted. And different markers, both surface markers and internal markers, can be used to investigate that state of the cell. Notably, PD-1, CTLA4, and LAG3 are garnering a lot of attention in the research community right now because they are indicators of cell exhaustion, which means that a T cell that expresses them is not currently active. Even if you see these cells, for example, within a tumor, they're not going to attack the tumor. And the underlying theme here is that using these markers, we can learn both what a cell is and what a cell is doing or going to do. Flow and mass cytometry are two technologies that allow us to measure the presence or absence of different cell markers in single cell resolution. Briefly, the way flow cytometry works is we take our cells, denoted as circles here, and we use fluorescent tagged antibodies to identify the different markers. So the cells are going to fluoresce in different colors based on the expression level of different markers. We run these cells through a tube, shine a laser on them, and measure the light that they reflect. In this example, different lights will be proxies for different markers. And again, the bridge between the fluorescence and the marker are antibody molecules that bind to the target markers. From a logical standpoint, the output from flow and mass cytometry are big tables of numbers, where every row is one cell, every column is one of the markers that we're measuring. It is quite common to see these kinds of figures that visualize this data in 2 dimensions. These figures are called scatter plots or biaxial plots. Each one of these dots is one of the cells, and each axis is the level of a different marker. Now it's really important to remember that we're measuring through several layers of proxies. The values we see here are the intensity of the light that we measure. The intensity of the light corresponds to the abundance of the antibody, and the antibody binds to the marker. So there's always noise around these measurements. However, they are good enough to be able to say certain things. For example, we can say that these cells over here express CD14 and MCSF receptor, while the cells in this quadrant express CD14 but do not express MCSF receptor, while these cells are negative for both of these markers. Now, here is a bit of a catch. The researcher decides which antibodies to include in their experiment, and this set of antibodies is called the panel. In other words, when we run a flow on the or mass cytometry experiment, we're going to measure a certain set of markers. We choose these in advance, and these are the only markers that we measure in a given experiment. Thankfully, there's quite an extensive library of literature that covers different panels and different markers, so there is a significant amount of guidance out there that allows you to choose the markers that are relevant for your experiment. With that said, there is still a bit of an art in building the panel and optimizing it. This is a notable example from a few years ago that proposes a set of 8 color panels that target different compartments in the immune system. So these 3 panels are each helpful for delineating T cell populations. This panel here includes markers for B cells, and this last panel is quite broad in targeting dendritic cells, monocytes, and NK cells. So to summarize, here is what flow and mass cytometry allow us to do. We have different samples, in this figure denoted as rectangles. For each rectangle, we have a big table where every row is one cell and every column is the different markers that we measure. Some of these markers are going to help us to identify the cell type, and some are going to pertain to the cell state. Now, these markers sit on a high dimensional space. N is the number of antibodies that we include in the panel, so it's the number of markers that we're going to measure. And we can use these to identify the different cell subsets in a given sample and their state, for example, activation versus exhaustion. So we're going to end up with these tables where every column is now a sample and every row is a different cell subset. And we can measure the abundance, the frequencies of these cells. As a reminder, this is one of the components of immune monitoring we mentioned before. Another table is going to allow us to look at the different cell state markers, and these let us examine the state of the different cell subsets across the samples. In the next videos, I'm going to cover different statistical techniques that allow us to take these numbers and actually make sense out of them. In other words, we want to take this single cell data, we want to map it into logical values that have some meaning in the context of biology, and then take this context and tie it back into the different groups in our experiment. In order to make sure you don't miss the next video, please subscribe to us on LinkedIn and on YouTube. Links are going to be in the video description below. Join me for the next video where I'm going to talk about different analyses methods for high complexity cytometry in the context of immune monitoring. Thank you for your time and have a great day.